This is a video tutorial for curtainptm, the post-translational modification data analysis tool brought to you by researchers from the MRC Protein Phosphorylation and Ubiquitination Unit at the University of Dundee. This project is supported by the Aligning Science Across Parkinson's Research Network. Hello and welcome to the second tutorial for curtainptm, which will show you how to make the most of the features of this tool to learn about post-translational modifications in your data. So starting from the point where we have either submitted our own data or clicked on a link, when we scroll down we will see a volcano plot. And an explanation of the default appearance of the volcano plot and instructions for making any customizations can be found in the How to Customize and Export Plots in Curtain video. So let's search for a protein, syntaxin 12, click search. And you can see now that when it's selected in the volcano plot, multiple data points are selected because these correspond to the multiple different modified peptides that are derived from that particular protein, unlike in curtain where one protein would just highlight one data point. So you can scroll down and see the information that has appeared for this protein. So this is the raw data distribution tab. And you can see that there are four modifications for this protein at two different serine residues, 139 and 142, but these are on the same peptide because there's the same peptide sequence here. So if you want to see the raw data distribution for a particular modified peptide, you can go to Actions and toggle bar charts, and these plots will appear below. So the bar chart, the combined bar chart and the violin plot, and you can disappear them by clicking on toggle bar charts again. And if we go to the PTM position viewer tab, we see a visualization of the linear protein sequence, which is aligned with the UniProt sequence by default, but we can compare against a different database, for example Phosphocyte Plus, by clicking on it here and selecting redraw plot. And now you can see that this has appeared below as well. So let's zoom in this section. So you can see the two modified serines, 139 and 142. And these are both in green because these are the selected sites. And they have asterisks above them because these are the sites that are changing significantly between experimental conditions. You'll notice that now that we are zoomed in, you can see the numbering for all the residues in the amino acid sequence, which is quite helpful. So in the UniProt alignment, serine 139 and 142 are in purple because there is an overlap between the PTM in the experimental data and in their database, and the same is true for Phosphocyte Plus. But now if we go back to auto scale to set the axes, you can see that there are some residues here in blue. So these have come up in blue because while they are reported in the database, UniProt and Phosphocyte Plus has an additional one that is not reported in the UniProt database, but they're blue because they are not found in the experimental data. Go back to auto scale again. So we don't see this here, but if any residues were to come up in red in the experimental data, that would indicate that that PTM was not observed in any of the selected databases. So you'd possibly found a new PTM. So scrolling down, you can click on kinase library here to see a list of predicted kinases that may phosphorylate this site. And you'll see that there is a web link at the top which will take you to the kinase library website. So one thing to remember when using curtainptm is that the residue numbering in your search output, so this here, may differ to that reported by the databases because the databases will be based on the canonical sequence, which is usually the longest isoform, whereas the search algorithm, so MaxQuantPTM in this case, may have selected an alternative shorter splice variant. So just to illustrate, we can see that this has happened in the case of a protein called NEDD4L, which I'll just search for. Click search to select it. There we are. So if we go to the PTM position viewer for this protein, we can see in the table below that there is a mismatch between the residue numbering for the experimental data and for UniProt, but we can see that curtainptm has still aligned these residues correctly, despite the numbering being different. So the last feature I want to highlight is the web logo feature. So I'm just going to clear my selections, redraw plot. So the web logo feature is helpful if you have a group of peptides selected that are substrates of the same kinase and you'd like to see if there is a common consensus motif. So just to show you how to do this, if I go to batch search and I'm going to copy and paste in a list of modified peptides here, which are substrates of a kinase called SGK3. So I'll just give this list a title, and the identifier type is not gene names. I need to change this to primary IDs, because these are the unique identifiers which we set as primary IDs in this session. So I'll click submit, and you can see now that these substrates have been selected in orange. And then if I go to Volcano Plot Actions, Web Logo Viewer, this web logo graphic appears. So we can see the phosphorylated serine appear here, with arginines at the -3 and the -5 position. So you can see this consensus sequence quite nicely here. So thank you very much for watching this tutorial. I hope it was helpful and that it encourages you to give curtainptm a try.